Good morning, everyone. Thank you so much for joining me today. I'm really excited to share with you a deep dive into a fascinating study published in Nature in 2020 by Daniston titled Structure of LRRK2 in Parkinson's Disease and Model for Microtubule Interaction. This work represents a huge leap forward in our understanding of molecular mechanisms underlying Parkinson's disease, particularly as they relate to the protein LRRK2 or leucine rich repeat kinase 2. My talk today will guide you through the motivations behind the study, the structural biology techniques used, the key findings, and the broader implications, not only for our understanding of disease, but also for drug development. So let's begin by setting the stage with some background on Parkinson's disease itself. So Parkinson's disease is a chronic, progressive neurodegenerative disorder that affects more than 10 million people worldwide. It's primarily known for its motor symptoms, things like tremor, rigidity, bradykinesia, or slowness of movement. These symptoms result mainly from the degeneration of dopaminergic neurons in the substantia nigra, a region of the midbrain that plays a central role in movement regulation. But Parkinson's isn't just about motor control. There are also non-motor symptoms, including sleep disturbances, depression, cognitive changes, and autonomic dysfunction of all, which can significantly impact quality of life. At a cellular level, one of the most well-known pathological hallmarks of Parkinson's is the presence of Lewy bodies, which are abnormal aggregates of the protein alpha-synuclein. These aggregates are found in neurons and are thought to contribute to cell death and disease progression. While the majority of Parkinson's cases are sporadic, about 10% are familial, caused by mutations in specific genes. One of the most commonly mutated genes in familial Parkinson's is LRRK2. LRRK2 is a very large multifunctional protein. It contains both kinase and GTPase domains, which is somewhat unusual. These domains allow it to carry out enzymatic functions that are tightly regulated in space and time within the cell. In addition to these catalytic regions, the protein has a number of scaffolding domains that likely help it to interact with other proteins and with cellular structures like membranes or cytoskeleton. So now several mutations in LRRK2 have been linked to Parkinson's disease. The most well known is G2019S, which increases the kinase activity of the protein. Others include R1441C/G in ROC domain and Y1699C in the CORB domain. And these mutations are generally thought to be gain of function, meaning they enhance or change the activity of the protein in a way that's harmful to neurons. A major focus in LRRK2 research in recent years has been on how it interacts with microtubules. Microtubules are dynamic polymers made of tubulin that form part of the cytoskeleton. They play a crucial role in intracellular transport, providing tracks for motor proteins like kinesin and dynein, which carry cargo such as organelles, vesicles, and protein complexes. Disrupting these transport systems can have devastating effects on neurons, which are highly dependent on long-range transport for survival and function. And in previous studies has shown that LRRK2 can bind to microtubules, in some cases form filamentous structures along them. And this filament seems to interfere with the movement of motor proteins, suggesting the possible mechanisms by which LRRK2 mutations might impair intracellular transport. But we didn't know the structural details and we didn't understand how LRRK2 was assembling on microtubules and how its conformation, particularly that one of the kinase domain, might regulate this assembly. And this is where the study comes in. So, authors set out to determine a high resolution structure of the catalytic core of human LRRK2 to study how it interacts with microtubules and how it explores how different conformational states affect its function and its ability to form filaments. To do this, they engineered a fragment of the human protein spanning residues 1327 to 2527. And this includes the ROC, COR, kinase, and WD40 domains, the key enzymatic and structural components of the protein. The researchers focused on the critical regions of LRRK2 protein and what they call as LRRK2 RCKW. This segment includes four major domains, the ROC, COR, kinase, and WD40. And again, as I stated before, they're spanning the residues are from 1327 to 2527. And this catalytic half of the protein was cloned into baculovirus expression system for high-yield protein production in SF9 insect cells. They used PCR to amplify the DNA, then inserted it into the PFB6HZB vector through the ligation-independent cloning. And backmid DNA was generated using the back-to-back system. For experiments in mammalian cells, they used the P-Des53 vector to express both wild type and in the Parkinson's associated I2020T mutant tagged with GFP. To support the downstream assays, additional constructs were prepared for proteins like kinesin and ninine used in modern protein experiments. Next, LRRK2 RCKW was purified through a multi-step protocol. Initially, the protein carried an N-terminal histidine 6Z tag for affinity purification, which could be cleaved using the TEV protease. The SF9 cell lysates were processed using NINTA affinity chromatography, followed by SP Sephiroth ion exchange. After tag cleavage, second purification round ensured removal of contaminants. Finally, they used size exclusion chromatography or SEC to polish the sample in high salt buffer containing GDP. This not only ensured stability but yielded in 1.2 micrograms of protein per liter of cell of culture, sufficient for higher resolution structural work. To determine the oligometric state in solution, they used SECMOS, or multi-angle light scattering, and this revealed that LRRK2 RCKW is mainly monomeric, with some dimers forming especially at higher concentrations. With protein in hand, the team moved on cryo-electron microscopy. For the trimmer data, they concentrated LRRK2 RCKW to 4 micromolar or more and applied it to glow discharged gold grinds. And these grinds were the plunge frozen in liquid ethane. Imaging was carried out using a Titan Cryos microscope equipped with the K2 Summit direct electron detector and quantum energy filter. And over 3,800 movies were collected in counting mode, yielding very high resolution images. They also prepared monomeric and dimeric samples, some treated with MLI2 or ponatinib to investigate kinase conformation under using conditions. And these samples were imaged on Talos Arctica microscope at 200 kilovolts. For microtubule-bound LRRK2 filaments, they polymerize bovine tubulin in the presence of LRRK2 RCKW. The protein was added in a 2 to 1 molar ratio to tubulin under low salt conditions to promote binding. These mixtures were also plunge frozen and imaged using cryo-electron microscopy. The cryo-EM data was processed using the combination of motion core 2 for alignment and the GCTF for contrast transfer estimation. Particle picking was done with fine EM and Crayola, and classification and reconstructions were carried out in Reliant and CryoSpark. For the trimmer dataset, they achieved an impressive 3.47 alpha resolution using C3 symmetry. A second reconstruction using signal obstruction and focused refinement improved the ROC and CORA domains, producing 3.8 alpha map. And these two maps were combined to generate full atomic model. Model building was done using Rosetta Qt. Homology templates were chosen by HHPRED and with the ROC and COR domains modeled after bacterial Rocco proteins, and kinase and WD40 domains built from top-scoring homologous structures. Side chains in low-density regions were replaced with polyalanine, and GDP magnesium was added to the ROC domain based on structural alignment with known GDP ROC structure. A major theme of the study was understanding how kinase conformation affects filament formation. And to model a closed kinase state, the LRRK2 kinase domain was split at the junction of its N and C lobes. Each lobe was aligned to corresponding parts of the interleukin 2 inducible T cell kinase, or ITK, um, known to exist in a closed state. And this model reveals steric clashes in when fitting an open conformation into cryo-ET density map of LRRK2 filaments. To understand how LRRK2 forms filaments on microtubules, the team used integrative modeling. And so they began by docking the synthetic WD40 dimer based on the crystal structure onto 14 alpha cryo ET map. Then they added 4 copies of LRRK2 RCKW structure by aligning their WD40 domains. Both open and closed conformations were tested, and the closed conformation provided a better fit, suggesting that the filament's formation is favored by a closed kinase. They also examined the backbone clashes at the COR interface using Chimera and found that the closed model revolves more than 80% of them. Explore the functional consequences of LRRK2 filamentation. Authors in the paper used single molecule motility assays. They tracked the movements of fluorescently labeled kinesin and dynein complexes on microtubules in the presence of LRRK2 RCKW. Even at nanomolar concentrations, LRRK2 RCKW significantly inhibited both motors, acting as roadblock. Kinesin run lengths dropped while velocity remained unchanged, indicating steric hydrance rather than motor malfunction. Dynein was also blocked, a rare feature for microtubule-associated proteins. And interestingly, in Parkinson's linked I2020T mutant of LRRK2 RCKW blocked motors just as effectively, even though it forms longer range filaments. And so the team then tested whether kinase conformation could modulate this inhibition. So they compared type 1 kinase inhibitors like MLI2 and LRRK2 IN1 with the type 2 inhibitors such as ponatinib or the GZD824. And type 2 inhibitors, which stabilize the open kinase conformation, rescued motor motility, and reduced filaments formation. In contrast, type 1 inhibitors, which favor the closed state, enhance filament formation and increase motor inhibition. These results were confirmed in cell-based assays, and in 293 T cells. Treatment with JZD824 reduced LRRK2 filament formation, while MLI2 had the opposite effect. And so here are the results of their findings. So the construct of LRRK2 RCKW encompasses residues, as we mentioned before, 1327, 2527, and includes the ROC, COR, kinase, and WD40 domains. The cryo-EM structure was determined at 3.5 A resolution using trimmer configuration of LRRK2 RCKW formed on cryo-EM grains. Although this trimeric state likely results from sample preparation rather than physiological assembly, it enabled high-resolution visualization of domain architecture. The structure revealed an overall J-shaped conformation with the ROC and CORA domains flexibly arranged and closely associated with the C-lobe of kinase. This spatial arrangement hints at mechanism for a GTPase kinase crosstalk, an important feature of LRRK2 function. The end lobe of kinase domains extensively interacts with CORB domain, occupying a position analogous to that of cyclin A in CDK2 cyclin complexes. This interface likely stabilizes the kinase and integrates regulatory signals from GTPase domains. One notable discovery was unique 28 amino acid alpha helix at the extreme C terminus of LRRK2, positioned after WD40 domain. This helix makes extensive interactions with the kinase domain, suggesting a role in structural integrity or regulation, or regulation. So importantly, this region includes a known phosphorylation site, which may modulate kinase activity through its proximity to active site. A kinase itself was captured in open inactive conformation with disordered activation loop. Parkinson's disease mutations such as G2019S and I2020T reside with the loop or at interdomain interfaces, underscoring their potential to destabilize regulatory architecture and promote pathogenic behavior. A central question in Parkinson's disease research is how LRRK2 associates with microtubules. To investigate this, the paper built an atomic model of LRRK2 filaments using their high-resolution LRRK2 RCKW structure in previously obtained 14 alpha cryo-ET map of microtubule-associated LRRK2 filaments. Experimental cryo-EM images of purified microtubules incubated with wild-type or I2020T mutant LRRK2 RCKW showed that both proteins directly bind microtubules and form ordered filaments. The diffraction patterns indicated the I2020T mutants exhibited longer range order, consistent with increased filament stability observed in cellular contexts. Feeding the cryo-EM structure of LRRK2 RCKW into the cryo-ET map revealed structural clashes at the COR interface when the kinase was in an open conformation. However, when the authors modeled closed kinase conformation, These clashes were largely resolved. Over 80% of the backbone clashes disappeared. And this supports the hypothesis that the closed kinase conformations promote filament assembly, while open conformations disrupt this interaction. Filament modeling identified two key interfaces mediating oligomerization at one at WD40 domain and another at CORA and CORB domains. And these structures were verified through cryo-EM reconstructions of dimers formed in vitro, even in the absence of microtubules. Aligning these dimers in silico generated right-handed helical filaments resembling those seen in cellular cryo-ET images, suggesting that filament formation is an intrinsic property of LRRK2 catalytic core. Importantly, this microtubule does not seem to make residue-specific contact with LRRK2. Instead, it acts as a scaffold with acidic surfaces that align with basic patches on the LRRK2 filament, thereby enabling electrostatic-driven oligomerization. Given its localization on microtubules, the study next explored the effect of LRRK2-RCKW on microtubule-based transport. In a single molecule in vitro motility assays, LRRK2-RCKW was found to potentially inhibit the movement of both kinesin 1 and cytoplasmic dynein 1. At just 25 nanomolar LRRK2-RCKW, motor mobility was nearly completely blocked. Mechanistically, LRRK2-RCKW appears to function as a roadblock. Kinesin run lengths were significantly shortened, though their velocity remained stable, hallmarks of physical obstruction. Interestingly, unlike other microtubule-associated protein, as for example tau, that inhibit only kinesin. LRRK2 also inhibited dynein, likely due to its filamentous structures, which may prevent dynein from sidestepping around obstacles. An I-2020 T mutant exhibited similar inhibitory effects as wild-type LRRK2-RCKW despite its enhanced microtubule binding and filament order. This suggests that inhibition is not strictly filament length dependent, or that single molecule assays is not sensitive to subtle differences in filament dynamics. These results support a model in which LRRK2 oligomers on microtubules impede motor driven cargo trafficking, which could in turn affect intracellular transport of organelles and vesicles, processes known to be disrupted in Parkinson's disease. And building on the idea that kinase conformation regulates filament formation, the authors tested a panel of kinase inhibitors to modulate LRRK2 behavior. Type 1 inhibitors are known to stabilize the closed conformation, whereas type 2 inhibitors promote an open, inactive kinase conformation. As predicted, type 2 inhibitors rescued motor motility, allowing both kinesin and dynein to transverse microtubules in the presence of LRRK2 RCKW. Conversely, type 1 inhibitors enhanced LRRK2 RCKW's inhibitory activity, likely by stabilizing the closed state and promoting filament assembly. In cell-based assays, this inhibitory had complementary effects on LRRK2 filament formation, and overexpressed LRRK2 formed abundant filament that were suppressed by GZD824, but increased by MLI2. And this effects occurred without significant changes to total LRRK2 levels or microtubule architecture, reinforcing the role of conformation in governing LRRK2 filaments forming propensity. The implications for drug developments are profound, and inhibitors designed to suppress LRRK2's kinase activity must consider structural consequences beyond simple enzyme inhibition. And drugs that stabilize The closed kinase conformation may inadvertently promote toxic filament formation on microtubules, exacerbating transport defects. In contrast, open state stabilizers may offer a dual benefit: kinase inhibition and prevention of filament mediated motor blockade. And the last section is discussion. And so for the field of Parkinson's disease research, these findings are incredibly valuable. They help explain how specific mutations in LRRK2 contribute to disease. They also offer a roadmap for developing better therapeutics, drugs that inhibit the harmful activity of LRRK2 without interfering with normal cellular processes. More broadly, this study is a great example of how structural biology can inform our understanding of disease mechanisms. By combining cryo-EM with biochemical assays and cell-based studies, authors were able to connect atomic level structure to cellular functions and dysfunction. This kind of integrative approach is likely to become increasingly important as we work to understand other complex diseases. And thank you all so much for your attention. Thank you for listening to this presentation. And I hope this has given you a clear and thorough overview of this paper and why this is such an exciting contribution to Parkinson's disease research. Thank you again.